It's my great pleasure to introduce Silvia Frey, who is group leader at the Curie Institute, and she has worked for many, many, many years on stem cells, first in the gut. Actually, even before that, she was working on fruit flies, but we won't go back quite that far. And then in the mammary system as well. And I followed her work very closely during my time in the same department. And she developed some of the tools that have really been, I would say, essential in understanding, in particular, how Notch is involved. But I think today she's going to talk to us about some of the work that she's done in recent years, and in fact highlighting, I would say, just how little we know about mammary stem cells and lineage commitment, and the whole notion of plasticity during development, and then in the adult, as the mammary gland is a highly dynamic organ, as we all know, or at least women know. So thank you, Silvia. It's wonderful to have you, and looking forward to your talk.
So I would like to start with warmly thanking Edith for making this symposium happen. I didn't believe we would be able to speak in physical presence so soon, so to speak. And we all knew or suspected that Edith could make miracles. Now she just proved that she could. So thank you so much, Edith, really. I'm fed up of Zoom meetings, so I'm very, very happy to be here in presence. Okay, so following up on the thread that Laia started, I decided today to tell you about lineage commitment and plasticity of mammary stem cells. And so indeed, in the lab, we are trying to understand how you make a mammary gland, starting from the very beginning, so from this ball of cells that we call the embryonic mammary bud, where we don't know where mammary stem cells are located, nor how they behave dynamically. And we don't know what is their lineage potential. So are they multipotent, unipotent? And mechanistically, we're trying to also identify the signals that dictate their state and their differentiation potential. In addition, another outstanding question that we and many labs in the field want to address is what makes the mammary stem cell niche, because differently from the intestine that we also study in the lab, as Edith mentioned, there is no geographical stem cell zone like the intestinal crypt that Hans presented you this morning. And of course, we're interested, just like Laia presented before, to understand how you go from adult mammary stem cells to mammary tumors and breast cancer. So okay, so thanks to Laia. I won't take much time on the introduction. She already told you that the mammary epithelium is a bilayered epithelium with basal cells here in green that are outside, and these cells are also called myoepithelial cells because in addition to a whole set of basal cytokeratins like K5 and K14, which are shared with the skin, they also express smooth muscle actin, and this gives them contractile capacity to squeeze the milk through the duct at lactation. And the inner layer, it's composed of highly polarized epithelial cells called luminal cells. They're here in yellow, and they express a whole different set of cytokeratins that we call the luminal cytokeratin, and they also express the receptors for the hormones estrogen and progesterone. So in terms of mammary stem cells, I want to present to you a seminal study that came from the lab of Jane Wiesvader in Melbourne, where about 15 years ago, they actually separated by FACS these two cell types that I just told you about, luminal and basal cells, and they probed their capacity of reconstitute a functional mammary gland when transplanted in host mice that were deprived of the endogenous epithelium. And this is a very powerful functional stem cell assay we have because we can actually probe, as I said, the ability of cells to make a functional gland that can actually produce milk at lactation. And by doing these experiments, they found out that basal cells could reconstitute a gland, and luminal cells could not. And so this brought to the conclusion that mammary stem cells exist, and they lay within the basal cell layer. Now, as I told you, this was 2006, so it was about 15 years ago. And since then, the work of many, many labs, of here I'm listing actually the major contributors, realized that multipotency is a quality of mammary embryonic stem cells, and postnatally, the gland, this epithelium, is maintained by unipotent progenitors that are either for basal cells or for luminal. There are actually two kinds of luminal cells that either express or don't the ER and PR hormone receptors. And so this cellular hierarchy has been defined by lineage tracing that allowed us so to visualize stem cell behavior in vivo, but also in situ, so without removing the stem cells from their physiological niche. And this, as you will see, it's particularly critical. And so in the lab, we are actually using a lot lineage tracing techniques, and we developed inducible notch-expressing Cree lines that allow us, when crossed to reporter mice, to label genetically the cells that express any of the notch receptors, and then study the clonal dynamics of these cells and what kind of progeny they produce. And by using these mice to look at the mammary gland, we saw that the notch receptors are all expressed in luminal cells, but not in the same cells. Indeed, for example, with the notch1 receptor, we could label here, labeled by membrane GFP, we could label highly actively dividing cells that are quantified here with a Ki67 proliferation marker, whereas notch3 positive cells, you can see one in this section here in green, are more rare cells that are actually quiescent. And what was striking about notch1 expressing cells is that they were invariably negative for the expression of the nuclear hormone receptor, estrogen, and progesterone. And so when we analyzed the transcriptional profile of notch1 expressing labeled cells, in bulk in this case, we found a signature of unipotent luminal progenitors. And what was surprising is that this transcriptional signature is completely conserved in the daughter cells from these progenitors, suggesting a certain degree of cellular memory, so that the mother cells, they divide and give rise to daughters that are identical to themselves. The book definition of self-renewal, if you want. But what was even more interesting for us was that we found out that these cells were also highly plastic. And what I mean by that, here I'm just showing you again that notch1 expressing cells are luminal, so they co-express luminal keratins like K8. They never express basal keratin here in red like cytokeratin 5. But when we probe these cells by isolating them from their original gland, and we probe them in the transplantation assay that I just showed you before, we found that now these cells have a high regenerative capacity, and they present multipotency because they could make basal cells as well as luminal ER positive and PR positive cells, which they never make in physiological conditions. And so this told us that with transplantation, we can actually kind of reprogram the cells, reactivating a memory that comes from when they were in the embryo, when they were truly multipotent. And we believe that this is also what happens in cancer, at least in breast cancer. I'm showing you here two important studies from the lab of Cédric Blancpain and Momo Bentiris in Basel, where they basically expressed an oncogenic form of PI3 kinase, which is a very common mutation in breast cancer, in adult unipotent basal or luminal progenitors. And just by doing so, they could reactivate multipotency signatures and generate tumors that co-express basal and luminal markers. And this co-expression of our known markers for basal and luminal cells is exactly what we see in the embryonic gland, as you will see in a minute. So basically, this is not a particularly novel concept. Actually, if you go back in history, in 1838, Johannes Müller proposed that cancer derives from reactivation of embryonic programs, and this is the basis of what we call the stem cell origin of cancer theory. And indeed, there are a few a subset of papers, particularly in the mammary gland field, that found strong similarities between the signatures of embryonic mammary cells and human breast cancer. So inevitably, we were drawn into looking at the embryonic gland, and so we started by doing lineage tracing in the embryo. So we would label embryonic mammary cells with our Notch1 Cree line, and then look at what kind of lineages these cells would produce. And when we did that, we found basal cells marked, as well as luminal cells, both ER positive and ER negative. So basically, we found that if we initiate tracing in the embryo, the Notch1 Cree line can label all different types of mammary cells. And this is the same line that I told you can only label unipotent luminal ER negative progenitors postnatally. And actually, we wondered if we were indeed marking multipotent stem cells in the embryo, or if we were labeling different types of already committed progenitors. And so we started looking more carefully in which cells express Notch1 in the embryonic gland. So by looking at 24 hours upon Cree induction, we would see the cells that actually express Notch. They're marked again by membrane GFP here. And just suffice it to say that Notch1 is expressed at all embryonic stages in cells that co-express both luminal and basal markers, just like every other embryonic cell at this point. And so then, to better understand the clonal dynamics of these cells, we moved to a multicolor reporter that actually Hans generated, and he presented this morning, the confetti reporter, that was kindly provided to us by Sharagim, so we're all interconnected today. But in any case, so basically this reporter allowed us to follow clonal lineages with different colors, and so follow clones derived from different mother stem cells. And by doing also some mathematical modeling with the help of Edouard Arnezo, who was in Ben Simon's lab in Cambridge at the time, now he has his own team in Vienna. We found evidence for unipotent stem cells already in the embryo. And indeed, actually, we calculated the difference between the real bipotency, what we would observe experimentally, and then we calculated statistically what would be the chance bipotency, so the possibility that we labeled with the same color, in the example shown here with red, two different unipotent committed progenitors, one that would give rise in one branch to a luminal clone, and the other one that would give rise in another branch to a red basal clone. And by doing this kind of statistical modeling, we found that actually at E15, there was, all the later time points from here, there was no statistical difference between the observed and the chance bipotency, suggesting that maybe already at E15, the mammary gland contains only committed unipotent progenitors. And the other thing that we are also interested in understanding is, of course, in order to commit to a defined lineages, cell receive input from external cues, but also from intrinsic signals. So it was easier for us to address if intrinsic signals could change cell fate. And to do that, we induced a sustained Notch activation using a Notch gain-of-function mutant that we've been using in the lab for the last 20 years, I think, in embryonic multipotent cells labeled by Notch1 Cree. And in this case, we noticed that we would actually, all the mutant cells would only give rise to luminal ER-negative cells. And so these experiments actually defined Notch1 as a master regulator of luminal differentiation, and it can force multipotent stem cells to give rise only to luminal cells. And so then we asked what happened if we do this in adult mice. I just told you in adult mice we cannot see any sign of multipotent stem cells. And we ectopically activated Notch in basal cells that never experienced Notch activity. And we did that using two basal Cree. Here I'm showing a SMA Cree, but we did it also with K5 Cree. And so these lines, they label 100% of basal unipotent cells. But when we put active Notch in it, we observed a fate switch, and now the labeled cells, mutant cells, they all became luminal. And what we observed was that if we follow this lineage conversion by FACS, so in a more quantitative way, because I told you we can separate a luminal and basal cell gate, we see that we start from labeled, these are the GFP, so mutant cells. We start from basal cells, and these cells, they gradually transit and move towards the luminal gate until by 6-week chase, they all became pretty much luminal. And so this is a progressive phenomenon. It doesn't happen just in one go. It clearly requires cell division. And what we are trying to understand is if this implies transitioning through, these cells in the middle, in between the two gates, through a multipotent stem cell state. And so indeed, we could quantify this also by co-expression of basal and luminal markers. So this would be the cells in green that we call the hybrid cells. We cannot define them as basal or luminal because they co-express all the markers we know of. And so we started analyzing their single cell profile by index sorting. So we used the SMART-seq approach in collaboration with Bertie Gotkins in Cambridge. And indeed, by differentiation pseudotime, we could see that the basal cells have to go through this hybrid state before switching to a luminal lineage. And the transcriptional program of the hybrid cells is actually very similar to the one of multipotent embryonic cells. And so here I'm going to open a short parenthesis to present you a very exciting story that we've been knowing since some time. This is not our story. It's from the lab of Cédric Blampin, and that just came out last year. And so what they did, instead of using mutants now in adult mice, they, through a pretty complicated genetic trick, they managed to lineage trace basal cells, adult unipotent basal cells that were labeled by tomato in red, and that normally give rise only to basal cells. But at the same time, the lineage ablated luminal cells using a diphtheria toxin approach. And so, whereas, as I said, in the control, these cells are strictly unipotent, after 2 weeks of losing luminal cells, what they observed is that these cells reacquire multipotency and start generating luminal red cells. And what was interesting is that they found that this is a reactivation of multipotency is conserved in other tissues, at least in other glandular epithelia, like the prostate and the salivary and sweat gland. And when they looked by single-cell RNA-seq, they found that the multipotent cell that they managed to reveal through this genetic trick, if you want, they have indeed a hybrid signature in between the luminal and the basal signature. So very similar, that has a lot of similarities with the embryonic multipotent cells. So basically what we're trying to understand now is really how mammary cell-fate-specific lineage commitment occurs and exactly when during the development from this round ball of cells to a very complex and beautifully branched epithelial tree. And we're doing this through two approaches that I'll briefly mention. One is a barcoding approach that we started doing in collaboration with Leila Perrier, who will talk this afternoon. Once again, we're all interlinked today. It was not on purpose, but it happened. And so, this is actually linked to also the definition of the single-cell transcription of embryonic mammary stem cells, both in wild-type and in mutant condition, to try to get both at the quantitative level the proportion as well as the transcriptional signatures of multipotent and unipotent stem cells that we know coexist in the same mammary embryonic bud. And so I'll just briefly mention how we are trying to go along with the lineage tracing by barcoding. So we're using mice that actually Leila developed during her postdoc that allow us through in a crene-deducible way to now mark cells not with a fluorescent color but with a genetic barcode. And we're using different luminal, basal, or even ubiquitous Cree to induce barcode writing in the embryonic mammary gland at different embryonic stages. And then we let these cells give rise to their clonal progeny. We separate luminal and basal cells through our FACS approach. And the idea is that if we find the same barcode in both luminal and basal gate, that would indicate that these cells derived from a multipotent mother cell, whereas if the same barcode is only found in one of the two gates, that would be an indication of unipotency. So I don't have results to show you yet about this, but I want to just take the last minutes to present you the second approach, which is the single-cell RNA-seq approach. And this is part of the project of a fantastic student that I'm really lucky to have in my team, Claudia Carabagna. And she is interested in understanding how lineage commitment is coordinated with branching morphogenesis in this embryonic time when there is a lot of tissue remodeling as well in this system. And so she's doing also a lot of imaging, but I won't talk about it today. What I want to just briefly present is her initial results in understanding which programs orchestrate the lineage restriction of mammary stem cells that I told you about. And for this, actually, she took another single-cell RNA-seq approach with the 10x genomics platform at Curie. And I'm just highlighting this because actually this was a pretty technically challenging experiment to get enough cells from these very tiny embryonic buds that you can see here highlighted in the LGR5 GFP model, because LGR5 labels mammary epithelial cells, and they're really tiny. So she had to dissect a lot of embryos. And then she sorted both epithelial and stromal cells. And she could define, with the help of a bioinformatician postdoc in the lab, Wenji Sun, as well as a fantastic master student, Robin Journeau, they could identify mammary epithelial cell clusters as well as the mammary mesenchymal cells, of which we know very little, but this might give us the famous external cues that also help cell fate specification, as well as a couple of skin cell clusters. And by RNA velocity, what they found is that in the embryonic gland harbors some, within the mammary epithelial clusters, they found an undifferentiated cluster that could represent the multipotent stem cells. We don't really know yet. And that can actually take two terminal endpoints and differentiate towards a luminal-like or a basal-like lineage. And this definition of luminal and basal-like lineage in the embryonic gland is really new, because as I told you, up to now, we cannot find anything that distinguishes them, and they all seem to co-express all the markers we know of. And so, of course, they found out the gene expression patterns that actually are changing along these two different differentiation trajectories, and this allowed Robin to pick two of his selected luminal and basal genes, and so he picked two favorite genes to generate some new reporter mice that we already started, we are in the process of generating, that would allow us to integrate the single-cell RNA-seq data with live imaging. And in this, so the genes we picked are supposed to be expressed in the gray dots. This would be the undifferentiated cells. So the idea is that you will have undifferentiated cells that would co-express both our reporter, fluorescent reporter, and then they would switch off one of the two and keep on the other one, depending if they go towards luminal or basal differentiation. At least this is the dream plan for Robin's PhD project. We hope it's going to work. And again, I mean, we can actually observe this by live imaging, so because the part that I don't have time to tell you about Claudia's project is being to actually follow by lineage tracing mammary stem cells that differentiate dynamically in time and space. And she could do that because she optimized the growth of embryonic explant cultures that can actually branch under a time-lapse microscopy. And at the same time, we are also made some progress in trying to follow what happens in terms of cell fate transition later on, postnatal in pubertal mice, by intraviral imaging. And for this, two postdocs in the lab, Guillaume Jacquemin and Bethan Lloyd-Lewis, developed a completely flexible imaging window that allows us to follow branching morphogenesis during the growth of the actual mouse body at puberty. And just anecdotally, I would like to say that the window will come up this week, and it's thanks to a very nice collaboration that we had with Chaire and Maria, who actually tested this window for intraviral imaging of the muscle, and it seemed to be working. So we managed to prove its more general utility. So thank you very much for this. Anyway, so just to conclude, we're trying to integrate the transcriptional circuitries that define unipotency and multipotency with analysis in real time, so in 3D plus time, to understand how we coordinate, at least in this tissue, cell differentiation with branching morphogenesis. And so we chose this tissue because, as I showed you, there is a clear progressive loss of multipotency, unlike, for example, in the gut, where stem cells stay multipotent for, throughout all our life. And so this allows us to study what triggers the exit from the multipotent state, but also what triggers the re-entry in cancer, and what signals actually control this adult cell plasticity. And with this, I will actually conclude by thanking, of course, all the colleagues. These are the more recent ones. Unfortunately, you can't see the faces. That actually, compose a fantastic team that I'm very happy to work with them, as well as, of course, the funding that allows us to do the experiment. And thank you for your attention.